Welcome back to Vaccine Design course. So here in this lecture, our step is to predict the T cell epitopes of the protein. So for that we need to go to the server called ProPred1 server. This is MHC class 1 peptide prediction server. So go to this particular link, ProPred1 or Google ProPred1, and click on the third result, which is MHC class 1 binding peptide prediction server. So this is the home page of it. Now to talk about MHC genes, they are expressed to produce surface antigens on cell membrane. So the main difference between MHC class 1 and class 2 is that MHC class 1 molecules present antigens to cytotoxic T cells with CD8 plus receptors, whereas MHC class 2 molecules, they present antigens to helper T cells with CD4 plus receptors. But basically MHC class 1 and 2, it comes under T cell epitope prediction. Okay, so this is your home page. So this is actually your glossary page where you see what the sequence, format, the various interpretation of your results. Okay, let's go to the home page. Just click home button here, and then here you can see you have to paste your sequence. So paste your particular sequence, Control A, Control C, paste your sequence. So this is in FASTA format. So make it as standard sequence format. Now for multiple selection use, you can, this is for allele selection. So here you can actually select your certain alleles if you want, or if you want to select all the alleles, you don't have to go for all like this. You can actually select the all button. Okay, now the result mode, that is the output display mode, you have HTML1, 2 and graphical, right? So here can you see the help button here? Just go to the help button and see the differences between the HTML1, 2 and the graphical format. I would prefer you to select the HTML1. Okay, so basically these sub options, they are useful for viewing the predicted overlapping binders. So why I prefer HTML1 is the overlapping regions, they are presented on separate lines, making it easier for us to detect the overlaps. Okay, so yeah, anyway, read this help page properly, which says about the server, then all the other algorithms, the thresholds, and again about the various output mode and everything. Okay, yeah, then yeah, okay, so here I'm making everything as default, making the threshold as 4%. Tabular results, I want 4 peptides to be displayed. If you want more number, you can do that. Then if you want to keep on the proteasome filter, which filters their MHC binders who have standard proteasome of immunoproteasome cleavage sites. Okay, so yeah, by default it was off, right? You can put it on if you want. That basically depends on your research aim. And then after doing everything, you have to submit your sequence. Then after submitting, you'll get your results. As you can see, we haven't given any name for the antigen, so that's why it's written as untitled. Then you have your length of input sequences, you have the number of nanomers, threshold setting, it was 4%, right? The number of alleles, there were 47, and the filters were off, so that's why it's termed as off. So now the displays are there, it's in HTML1 format. If you want to go for HTML2, you can do the same procedure like that. Yeah, so these are the tabular display. Okay, so this is how the tabular display shows. So this, I'm going to the help button and see the interpretation of the results. But when we did the exercise, I haven't made it as HTML1, it was HTML2 result. So I've again made it as HTML1, and let's look. So this is how the HTML1 results are displayed. So you can actually do the HTML2 and see the difference between. This is why. So here can you see the overlapping regions being displayed as different lines, so that it's, like I said, it's easier to detect the overlaps. Okay, so this is how it's showing. As you can see that these peptides are represented more, or you can say it is expressed more than the simple string. That's how you have to interpret the result. You can take the help with the help of the help button, as I've mentioned. Can you see these are the examples that they have given where they've mentioned that this peptide, which is this, the RTFE, they are all represented more expressively than the simple RTFE. So these are the overlapping regions, okay, or the MHC binding regions. Now, this is one way of representation. When you scroll down, you can actually see two types of representation, which is graphical display and sorted binding regions, right? So click on the graphical display now. Now you can use this graphical display for your paper. You can cite this particular thing. Now this is how the graphical display looks like for the MHC class 1 binding peptide. So on the left hand side, if you click here, it will say how the graph looks like. So the x-axis represents the amino acid sequences. So this is your threshold value, and the y-axis is the peptide score. So if you go back, you can see the peptide score and the threshold value. On the right side, where it has beautifully represented the score and threshold profile, when you click here, it says how the graph looks like. It's from 0 to 10, right? And the red dotted lines indicate the user selected threshold, okay? And the y-axis is actually the number of frames, how many frames you have, and the x-axis represents the threshold, okay? Which is, so the 1 to 10 represents the number of frames predicted as binder, okay? So this is how it goes. So this is again the graphical display which better interpret the results, right? Next, when you go back and go for the other one, which was sorted binding regions, which says about the sequence, the position number, real score of each residues. So the first one means it's the rank 1. It's based on different ranks. The first one is the highest score. Okay. Now you can go to the help button and see how the tabular format, which is the most widely used option for results display, right? It displays the peptides sorted in descending order of their score. So this creates a separate table corresponding to each selected allele, okay? So these are for not residues, it's for each selected alleles. So now we looked at the graphical format. So here this mentions about the graphical display. Then you had the tabular format, and then you had the text or the HTML1 format, right? Yeah. So again, it says about the limitations as well. So please have a look at it. So this is how you interpret the MHC class 1 binding regions. So for MHC class 2, again go to ProPred. Okay, let's go to the PowerPoint and let me just show you the steps again. So yeah, we pasted the sequence, we selected the mode as HTML1. Now your exercise is to go to HTML2 and see the difference, okay? Then you will be able to identify that HTML1 is easier to interpret. So that's why. Okay, so for MHC class 2 binding peptide prediction, you need to again go to it was ProPred 1 for MHC class 1. Go to ProPred, the main site for MHC class 2. Okay, so then try the same with the same parameters we applied above and use according to your research aims. You can again see the graphical, the tabular format and everything. It's the same thing that it's been shown in the MHC class 1. Okay, you just have to again go to the help button for better understanding of the results. Now let's go to another server to predict again the MHC class 1 and class 2. So if the first server was ProPred, right? Now the second server that we have is the IEDB. Okay, so go to IEDB or tools.iedb.org and then go to the T-cell prediction site and let's select the MHC class 1 molecule. So this is the IEDB tool site. Here under T-cell epitopes, we are going to peptide binding to MHC class 1 molecules. Here you need to paste the sequence in FASTA format. Copy it and paste your FASTA sequence. Then your MHC source species, we want it as human. And the next one that you see when you go down is select the HLA allele reference set. So you can select the frequently occurring alleles as well as the HLA allele reference set. So let's keep the frequently occurring alleles as default. And now to select the HLA reference set, you need to click here the button, and then it automatically gives result of all the HLA references. Okay, you can actually delete this and enter manually as well. You can press here this button, then all the alleles will get deleted like this. So you can delete everything like this and upload manually the alleles as well as its length. If you want a specific length, you can specify your length as 8, 9, 10, or you can mention it as all lengths. Okay, so specify how much ever you want. So for now, let's just take 4 alleles and see how it shows. So again, since I've selected only frequently occurring alleles, it shows only frequently occurring alleles. Okay, you can actually do not select this, unselect this and select the reference set only. Okay, either options works fine. Then after selecting your alleles, sort peptides by, so I've mentioned it as predicted score descent, and output format, make it default. If you want to give your mail ID, you can give your mail ID, that's completely optional, and then click the submit button. So this is your results page where you can see the alleles name, sequence number, its start and end, the positions, then the sequence length, peptide score, and the percentile rank. So the percentile rank is also mentioned. You can see that it's been decreasing. Here you can't download this, but you can copy and paste this particular thing. Okay, so the rank, since we have mentioned it based on the descending order, it's mentioned like this. So the first ones are actually the alleles which have the you could say the MHC class 1 binding sites. Okay, now to predict the MHC class 2 binding epitopes, again go to the particular link. Let me go to the PowerPoint and show you. Okay, so this was MHC class 1, right? So again go to the IEDB tool section, and under T cell prediction go to MHC class 2 binding. Select the peptide binding to MHC class 2 molecules. Again paste your FASTA sequence and see if the FASTA format is detected on the bottom right corner. Even when you do the class 1 also, see if it says the FASTA format is detected. Okay, only then can you proceed it. Now here you can see that there are 3 options. In MHC class 1 there were only 2, right? Here you have you can select the alpha and beta chains separately. You can select the full HLA reference set, and you can select the 7 allele HLA reference set. So here you can select only 2 options from the 3. For the previous one you had 2 options where you could select both. Okay, so these are the 3 different parameters which can be selected based on your aim. And now if you want to manually add an allele, you can do that as well by selecting the option that is select. Let's select only the alpha and beta chain, and then you can select the desired alleles. Okay, 1, 2, 3, 5. Okay, so here the length is not mentioned on the right side as we saw on the class 1. It has a separate option for it. Okay, so let's in length, the default length is again 15. So one or multiple peptide length can be selected for the prediction. Now the available length options is from 11 to 30. Okay, so a list of peptides will be automatically generated for predictions by scanning input sequence with the user selected length. Okay, so let's select 15 to 30. Okay, 15 to 30. Okay, so the default was 12 to 18. Okay, now the sort peptides I'm sorting it by the adjusted rank. Email address, if you want, you can give. Now you can either select only the alpha and beta genes or select the full HLA reference and then delete manually, and then select only the desired ones. Okay, and then after that you need to click the submit button. Now after getting the results, see if the results are in jumbled format. Okay, if it's in jumbled, then you need to do it again until the results become organized like this. Now here it's actually based on IC50, means to interpret how much toxic the protein is. Okay, so here when you did it automatically, sometimes you might get jumbled results. Okay, so when you do it manually, you will get arranged result like how we got it now, so that you can interpret easily. Here in the results option, you can see the download result. You can also download the results, okay, and save it in your PC. Then now for your exercise, do the same step by selecting the 7 allele option and see how you get the results. Okay, so try different options. As I've said, you out of 3 you can select 2 options. Try different options and see how the results come. Okay, so now if you click on adjusted rank, so it's based on adjusted rank. Now if you click on percentile rank, it will automatically arrange according to the percentile rank. Okay, so here you see that low adjusted rank means they are good binders. Okay, so it's already been arranged it as a good binder which has low percentile or adjusted rank, which means all these alleles are good binders. Okay, now so this is how you need to interpret for the MHC class 2 binding under the IEDB. Again, in the same site of IEDB, select the T cell epitope prediction tool, TEPI tool. Okay, it's actually a combined one which predicts both the class 1 and class 2 MHC. But again, it's always one at a time. Okay, so it's basically these are the 3 different servers under IEDB where you can predict the MHC class 1 and class 2, and you also have the ProPred server as well. Okay, but let's go to the TEPI tool option now. So this is the TEPI tool server. Here again you need to paste your FASTA sequence. Okay, see again if the FASTA format is detected on the lower right corner, and again click the next button. Here again one at a time. First let's go for class 1 and then click the next option. Next is you need to specify your alleles. So by default it's the list of frequently occurring alleles. So let's go by this, but if you select from these list, and you have to select your alleles as well. Okay, so I have selected 1 2 3 4 5 alleles and then click next. Again, the peptides to be included. What type of peptides do you want? So what I want by now is apply default settings for high number of peptides. Okay, so if you want custom selection, you can set your own settings as well. By custom selection, you can, if you want to remove duplicate peptides, you can do it. Then you can actually arrange your peptide lengths to be considered in prediction. But here I am applying the default setting and then clicking the next button. Then the type of predicted peptides that I want, I want these selected peptide based on the predicted percentile rank. If you want based on the predicted IC50, you can go for it. Okay, so that entirely depends upon your research aim. Then click on next button. Then after that it shows the review of how much you have entered, and then if you want to give your mail ID, you can give it and then click the submit button. So the result that was displayed is based on the percentile rank. Okay, so if you select the percentile rank, you could see the higher number of percentile rank, it's arranged like this, and the lower number. So again, a lower percentile rank indicates higher affinity. Then again, if you want to predict your result based on IC50, you can again do the same procedure and then click make the prediction as based on the IC50. Okay, you can always go to the help button and see how beautifully they have mentioned all the steps, and then they have also written a separate heading for the interpretation of result as well. Okay, here the results is prediction, interpretation, everything. Okay, so yeah, so this is how the MHC class 1 and class 2 binding sites are predicted. You have the ProPred, IEDB, 2 servers for the MHC class 1 and class 2 or T-cell epitope prediction. That's about it. Thank you.